of last, because
Recording
that
in
is
progress.
a exciting new open access capacity development. And in the middle, I'm going to tell you about the African Bioimaging Consortium, which is a community network that I'm involved in. So it is worth, before we jump into that, sort of stopping to for a moment and actually asking the question, this is all fine and well, but if the challenges are so complicated, is it actually worth putting in all of this effort to develop microscopy capacity in Africa? And I'm just going to show you two, I think, fairly striking figures to sort of highlight the disparity between what is available and achievable in Africa and elsewhere in the world. So this first graph, the red bars show you a population, between the red and blue, it's a population scaled disease burden in Africa. And this is across infectious and non-communicable diseases. Sort of representative to world populations, you can see that Africa across all regions has a disproportionate disease burden per capita. And on the flip side, though, all those very small green bars at the bottom are the sort of representative research output coming from each of the regions in Africa. So for the proportion of challenges that Africa deals with, there is limited research output capacity. And this is another graph just showing here across Africa. Green, again, is a metric of disease burden per capita. And those red lines all over the graph actually show that there is research being done in Africa to address this disease burden. It's primarily collaborative with the global north. In this case, it's just highlighting collaborations with Europe. And it means that the research output, that blue shading, is attributed to European institutions. And most African researchers are then functioning in collaborative projects where they are not the driving force behind a lot of the research being done. I don't know how well it's coming up on Zoom, but if you can see some yellow lines going across the African continent, and that shows some internal collaborations, but they're definitely outweighed by the North-South collaboration networks. So there is capacity, there is potential. Currently, the distribution of attribution and resources is incredibly uneven. So to kind of address, I hope those graphs highlight, it is absolutely essential that we build capacity development in Africa. So obviously, one aspect is that it will improve equitability and diversity in research. If Africans are or other underrepresented regions are dealing with the majority of the disease burden and other challenges, they should have the opportunity to drive the research and contribute to the direction and the solutions that they're looking for. This means that it can target local needs as opposed to the global north's idea of what will solve the solutions. It's also important to realize that local access to advanced research technology confers ownership of that research to researchers anywhere in the world, in a way that if you're simply shipping samples off to collaborators, or we frequently have to get on a plane to access technology, you don't have the same sense of ownership of the research. And you also don't have that chance of going down the corridor or to the institution across the city to say, like, hey, I just want to take a look and explore and discover. It really hinders the pace of research. And it's important to acknowledge that all of these, this is not exclusive to microscopy. The capacity development that we're doing in microscopy at the moment is happening in parallel to initiatives driving research infrastructure investments in computational biology, in sequencing biology, single cell technology, spectroscopy, biomedical imaging. So this is really critical that we build research capacity in Africa in general. It's just absolutely worthwhile doing this in microscopy in parallel. So one more, I'm going to start with where all of this started, and also hopefully this drives home this point once more. In January of 2020, we were involved in running a workshop in Cape Town. And this came out the idea that there weren't a lot of microscopy training workshops easily accessible to African researchers. Even if they're hosted in Africa, they tend to be open to the rest of the world. And as is understandable, it's a nice reason to travel to a pretty space for a workshop and a holiday. But the cost means that a lot of Africans cannot do the same. So this workshop was targeted exclusively to African researchers and students. There was no cost barrier. So if researchers and students were accepted, all costs were paid for the participants' flights, accommodation, and registration. And they had access to representative African as well as international faculty. And this was a large collaboration between University of Cape Town, some internal institutions, and the Advanced Imaging Center in Geneva. And when we started this, we actually had no idea how it was going to go. It was a sufficiently large learning exercise that a paper was written about the experiences gained from running this first workshop. But a key thing is that for 24 places on the workshop, we had over 700 applications. And if my previous graphs didn't convince you of the need to develop capacity in Africa, I hope that this evidence of a drive to learn imaging and technology science in Africa convinces you that this is a worthwhile endeavor. So out of this workshop, aside from a Nature paper, which is definitely worthwhile reading, we had the opportunity to speak to a lot of participants of the workshop. And we learned that while the workshop is nice, as I mentioned, the researchers go home to a diversity of challenges that a workshop alone cannot address. One of them being they go home and very alone. So out of that workshop, and in the middle of the COVID pandemic, first shutdown, we got a group of workshop alumni as well as other African researchers together online, and out of discussions with them started the African Bioimaging Consortium. So this is a community-driven initiative with a key focus of uniting and empowering the African bioimaging community, serving as a central focus where initiatives can come together, information can be distributed, and we can promote research as well as collaborations within Africa. A key driver behind this is if we build this community, it is a very powerful advocacy and dialogue partner with funders, policymakers, institutions to help change not only access to the science itself, but all the policies around us for infrastructure development. So just as I said, we started March 2020 with, I think, 18 researchers online from eight different African countries, and sort of statistics from the end of last year, we're now about over 200 researchers from about 20 African countries involved, with a good representation of different career stages and researchers working in across the board of different research fields in biological sciences in Africa, and a good spread across a number of countries, sort of in a reasonable distribution of what you can expect based on population and research culture in the countries. Very excitingly, from January last year, we were funded by the Caron Jacobs initiative, expanding global access to bioimaging program, which has allowed us to continue a lot of our activities and build on what we're doing. So I'm going to run through very quickly what it is that we do and try and how we try and build the community and empower them. And the first part being actually very simple. We have a website. The very first activity we did was build a website as an idea, as a central point for our community, as a landing point for them to find information. And I'm going to take you through a few of the features on the website, because a key thing that we wanted to do was actually let the community know about the resources available to them already out there that we haven't had to build. It's just signposting essentially to them. And as more and more relevant opportunities and portals resources become available, this is a fairly dynamic page that gets updated regularly. We also have in development a number of programs and blogs to really highlight what is happening within the community as well. Another key thing, again, in terms of signposting and improving access to information for the community, is that we've partnered with a new program called the Microscopy DB, which has come out of Bioimaging North America as a initiative to try and minimize duplication of effort across all of these community networks that are all trying to aggregate and distribute information about all the opportunities and initiatives out there. So as in partnering with Microscopy DB, information about workshops, funding opportunities, jobs, other training opportunities get centralized and then get posted on various community portals, including ours, in a way that is filtered to what is relevant to each community. So we're fortunate to be one of the first partners for the Microscopy DB website. And we have this opportunities and announcement database then on our website, where we are posting sort of some manually and some with Microscopy DB, just opportunities that are relevant, accessible, free of cost, their sponsorship opportunities, whether they are in person or online. Another key thing that we're trying to do is increase visibility of the community to each other and the rest of the world. And this is through what we call the Microscopy in Africa platform, or the MAP. And this is a very simple database of researchers where they can see where researchers are, where equipment is, get a vague idea. The information is fairly thin, but for researchers to get an idea of who's working on what, where, to find speakers, to find collaborators, to find supervisors. And for many researchers, they don't have an online presence prior to this. Many facilities don't have an online presence with the exception of this. So this is critical to growing global awareness for what is actually in Africa. As we are a community initiative, obviously the big thing is getting the community to work together. So we have some working groups we have 2 working groups at the moment focusing on training and education and the infrastructure and access, which is looking at expanding that information about what is there on the continent. So our first working group is really interesting. They are developing a targeted and tiered training curricula for microscopists in Africa. So this is trying to make sure it's context relevant, accessible for as many researchers as possible, and in a way that can be distributed. And the plan is for this to then be developed into a train the trainers program. And I've had the great fortune of being here in Montreal for the past week and a bit, participating in a fantastic workshop run by Claire Brown. And from discussions around train the trainers, I'm leaving here with my head overflowing with ideas of how we can further build on these plans. So it's very exciting. And one of the benefits of being able to work together with researchers in this field across the world. Our second working group is working on, as I said, building essentially on this map and trying to get a much more detailed landscaping exercise. It is not trivial, just trying to reach people, but it's an ongoing effort. And then last but not least, we have the great fortune of, supported with our CCI funding, having an almost completely sponsored annual community meeting where researchers can get together in person. And we've had our first community meeting was October last year. We had some fantastic community dialogues with each other, with commercial partners, and a lot of exciting initiatives in the pipelines from there. I'm going to keep going. A key opportunity that we have at the moment that we're very excited about is what we've called the SWIFT Mobility Awards. These are mobility awards to allow researchers to travel within Africa to a different country to access microscopes that they don't have access to, either for pilot experiments. So we have no expectation of research output, except hopefully that they would know that they can pursue more microscopy together and perhaps write a grant. And then for collaboration grants, where we really encourage researchers to develop partnerships within the continent for research. We have an active call for this at the moment. So for any of the audience with colleagues within the African community, I encourage you to share this opportunity with them. As I said, we have a lot of exciting projects in the pipeline from recent community dialogues, ones that I want to highlight because they're this little light bulb in the top corner is reminding me that there are ways that the audience can get involved if they are interested. we have a number of initiatives where we could do with input from the global community. One is that we are developing some Ask the Expert fora, where we have researchers who have expressed that they need help with things like experimental design, not on a sort of day-to-day scale, but how to plan an imaging project to determine what sort of technology they need, and how they can describe those systems in an accurate way that if they are able to purchase a system, when it goes out to tender, vendors actually have a good idea of what they need. We've joked that we're calling this the anti-white elephant program, because we're trying to avoid unnecessary purchases of equipment that's not suited for the needs of the community. And then we've got communities interested in things like best practices that they just want to be able to ask people about. Linked with that, we've got some growing mentorship and subject networks such as ECRs and a few image analysts who just want to get connected with each other and perhaps have access to some mentorship from the global community. And what will be coming up shortly is some application writing workshops, where again, the community has indicated that they want help with how to write good applications, whether it be to access a workshop or other training initiatives, grants related to microscopy, or access mechanisms, such as to access infrastructure within Eurobioimaging, or the Advanced Imaging Center, or the African Microscopy Initiative that you'll hear about from me in a few slides. So if any of these sounds like things you'd like helping people with, feel free to get in touch. And just to acknowledge that a lot of what we do is in partnership with existing networks and initiatives, especially with initiatives already functioning within Africa to provide training, to provide access to existing infrastructure. Our aim is not to duplicate effort. We want to work with people to make them more sustainable and effective. And we've had incredible support from the global community and networks. And I have the amazing privilege of standing up and talking to people about this, but I do not operate in this community alone, not by a long shot. So this is just to acknowledge that we do have a leadership team. We have some amazing working group coordinators and an excellent scientific advisory board. And there's some contact details, but they'll come up on the last slide as well. So the next thing that I want to talk to you about is what's called the African Microscopy Initiative. And this is a very exciting new program. It's a multi-pronged approach to develop open access microscopy in Africa. And this is sort of ticking the box of the building access to infrastructure and to the technology, as well as some training. So there are three key components to this. The first that I'll touch on is the flagship component, which is an open access imaging center and project incubator. And that is very exciting. It's absolutely free to use for scientists from non-profit African institutions. So that confuses some researchers. The key there is that there are a lot of private universities in Africa, but if the research being carried out is not for profit, then they have access to this facility. Its access is through a peer-reviewed application process. And as mentioned, there's a bit of an incubation program that I will touch on quickly. So this is not a technology talk, so I'm going to just touch very briefly on what we have in the facility. We spent a lot of time, this is a brand new lab, we spent a lot of time thinking about what would be the best spread of technology to make accessible to researchers in a reliable and consistent manner. So we've opted to go with commercial systems to start with, and really running the gamut from wide field systems that allow researchers to work with monolayered cells, microorganisms, whether that's bacteria, parasites, even small organisms if they don't need 3D imaging, as well as a fantastic confocal system with spectral detection and ARIScan 2, a super resolution system, the Elyra 7, with both localization microscopy and SIM, so we can cover almost all of the demands for high resolution imaging. What we don't have is STED, as well as the Cell Discoverer, which is Zeiss's high content system, which means that we can support screening assays, as well as what's exciting with the system is that it's designed for extended life cell imaging of various formats. So we can also do extended life cell imaging of small organisms, for example, for developmental biology. And then there's a huge, we're aware of a lot of researchers doing a lot of histology within the continent. So we also have the TissueFacts slide scanner for both wide field and fluorescence imaging. As I mentioned, this is a brand new lab, and what I'm showing you here is a wide field photo of the most absolutely incredible facility. We had the great fortune of coming up with this African Microscopy Initiative essentially as our institution was building an extension to one of the wings. And after some discussion, we were offered the ground floor of the entire new building. So this is an enormous space. We deliberately overcatered for the space requirements for each of the systems, because aside from individual researchers accessing these systems, the space is designed for training. So we can and already have run workshops in this lab. And there's a central lab bench for sample preparation, training on sample preparation. And what you can't see at the back there is a very small tissue culture lab, so that researchers visiting the University of Cape Town don't have to beg for space in other active labs, but they have dedicated space, and we can dedicate it to whatever samples researchers are bringing in, whether it's mammalian, bacterial, yeast, or anything in between. This lab is not yet, but we're expecting in the next few weeks to also be spec'd up to BSL2 level. So we really have a lot of flexibility in terms of what can be brought in. And as I mentioned, it's based at the University of Cape Town. It's within my home institute, the Institute of Infectious Disease and Molecular Medicine. It sits just behind this little peak of the roof there. And what's very exciting about this lab, aside from the zero cost to use the equipment, is that we are running a visitors program where for researchers whose projects are accepted, 100% of travel and lodging for researchers from outside of Cape Town to visit the center is covered by the initiative. And that most of the costs of actually running the experiment in terms of reagents and consumables is also supported by the initiative. And that is with the very generous financial support of both the Bill and Melinda Gates Foundation, as well as the Caron Jacobs Initiative, who are 50-50 funders of this initiative. So we operate on a similar model to, for those of you who are familiar with it, the African Microscopy Initiative at the Advanced Imaging Center, at Genelia in the US. And from that, we have put a lot of thought into how to support visitors coming to the center. So prior even to application, we encourage researchers to contact us for a technical consultation, but certainly before they visit, there would be consultations in terms of making sure that the experimental plan is sound and we all know what's going to be done. We will work with researchers to make sure they have the appropriate ethics clearance to do what needs to be done, to bring in samples that they need to bring in, and help with travel arrangements, etc. While here, as I said, accommodation is covered, costs of the experiments are covered. It's a fully equipped lab, as well as access to, if we don't have it in the lab, we do have access to a lot of key equipment in neighboring labs within the institution. And they have dedicated time on any of the microscopes they've indicated needed for their project, while here no one else would be using the systems that they are using for a period of up to 4 weeks. Through the visitors program. And then there will be help for image processing and analysis, and hopefully preparation of publications or grants coming out of projects coming to Cape Town. So I mentioned that there's also a project incubator program. The idea behind that is if we identify high value projects that could benefit from additional visits to the imaging center, they are not limited to only coming for a few weeks. We can support multiple visits, as well as if the project grows beyond the systems that we have available, we can facilitate access to either other equipment within the area, so either within the institution or the facility that I run with my other HAP, the IDM microscopy platform, as well as the facility at nearby Stellenbosch. And we can also facilitate and support researchers visiting other high-end platforms, such as at the MBL. And going one step further to really connect visiting researchers to the global community is that we have formed an alliance of sister imaging centers with the Advanced Imaging Center in Tunelia, as well as the advanced, I've drawn a blank on what it's called, the imaging facility at Institut Pasteur in Montevideo. And of course, researchers are automatically connected to the African Bioimaging Consortium when they visit the Africa Microscopy Initiative as well. So I'm going to run through quickly the other aspects of the Africa Microscopy Initiative, because as I've said, it's not good enough to just give them a few times access to some microscopes in the southern tip of Africa, and then they have to go home and they don't have access to microscopes where they are. So a key component that we're also running is instrument placement programs. The idea behind this is a program of strategic redeployment of previously owned or pre-loved, I think is the term that's been used, microscopes. And this is following a needs-based application process. We envision that this will run primarily for researchers who have already visited the facility, and we've identified that they could use this microscope well back home. And the idea with this is that these would form seeds of core facilities so that there would be an infrastructure network growing across the continent. So I'm going to pause here and do a little detour into a brief discussion around research equipment donation, not only in Africa, but anywhere. And it has its pros and cons. So on the one hand, it has incredible potential to really support research, form the seed of infrastructure and advanced research in an under-resourced setting, and really it makes donors feel good. It's an environmentally friendly way of dealing with equipment. It feels sustainable. But if done badly, it can lead to a lot of frustration and demotivation for researchers. If equipment that is not fully functional and has not been verified to be fully functional before and after delivery and installation, if it works for a while, cannot be serviced and then breaks, or if the one or two trained users or who are meant to help other users use that equipment then leave, what the receiving lab is left with is a very expensive piece of equipment that cannot be used. And frustratingly, it leads to a cycle of frustration and demotivation, not only for the researchers, but for institutions and funders, because what they see is they went through a lot of effort to facilitate delivery of equipment. Even if equipment is donated, there are transport costs and income taxes that often have to be paid, custom taxes that often have to be paid. And if they see that equipment not used, they're unlikely to go through that effort in future. And funders, if they see, well, equipment has been donated, it hasn't been used, and we don't see development of research around the technology, they are unlikely to support applications requesting access to similar technology in the future. So we have the possibility of either a virtuous cycle, that's really supporting and developing research, or a cycle of demotivation and frustration. With all of that to say that it's really important to be very careful about how we go about donating equipment to under-resourced areas so that it really supports rather than frustrates local researchers and communities that left feeling like dumping grounds for the rest of the world. So with that, what we have come up with and are planning to launch later this year is what's called the Program for Equipment Exchange and Reutilization. And this has come out of a few commercial partners indicating that they do have equipment that has been swapped out, for example, for when well-resourced labs are able to upgrade systems, or that labs no longer want to use systems and want to make them available, there is available of instruments. So the idea with the program is that when we have a few of these systems available, they will be announced. We ideally would have previously identified researchers and institutions that could make good use of them. And what we expect, and that's this little exclamation mark in the corner, is support from corporate and network partners. And what I mean by that is this is working with vendors, because we expect these corporate partners to make sure that the available equipment is working, will be serviced. So it's not out of service periods because it's a 10-year-old model and they're just no longer supporting it, and that it is functional, and that they will facilitate delivery and installation and validation of the system once installed in the recipient lab. And then we also expect the active participation and commitment of not only the receiving researcher, but the institution, because we expect institutions to commit to employing full-time staff to support the facility. So it's not just a lucky postdoc who moves on in 2 years' time, and that they can cover the cost of a highly subsidized or reduced cost service contract. And the idea, the hope with this is that this equipment is sustainable for a longer period of time, remains usable, and actually drives research, so that by the time it does pass out of service period with the vendors, the institution and the researchers have demonstrated capacity to do the work and can write grants to secure their own new equipment. Yeah. So the other exciting component of the African Microscopy Initiative is the training programs. These are continental and regional workshops, as well as a train the trainer program. So this will work very complementary to the Africa Bioimaging Consortium, or ABIC. And the idea behind this is, aside from building capacity, it's also building partnerships with the global network. So you've already heard about one of them. That's the Imaging Africa program that we have already run twice now in Cape Town. And the idea is to keep replicating this program every year, but it will start alternating between Cape Town and other cities across the African continent each year. And where necessary, it can be scaled down to become a satellite workshop with a thematic focus. For example, we're planning this year's workshop to be hosted in Dakar, Senegal in November this year. And they've indicated a key research focuses of Plasmodium and arbovirus research. And we will be training regional researchers on how to use available infrastructure to develop their own research. The second initiative with this is the Partners in Teaching program. The idea behind this is to build those regional partnerships. And again, there's a light bulb there because those interested can get involved in this. The idea is that there will be two pools, the pool of African applicants and international applicants. Envision this being primarily early career imaging scientists, or those who want to transition to be imaging scientists. And they will be paired, mentored with an international, more experienced imaging scientist and trainer, and supported in running workshops within the home region of the African applicant. And the idea here is, aside from running the workshops, there's building these relationships between the African and the international community, and everyone gets experience in running workshops in the process as well. So that, in a nutshell, is a very big nutshell, the African Microscopy Initiative. And just to acknowledge that it does not happen without the support of a lot of a number of key stakeholders and partners. Obviously, it's the African Microscopy Initiative, or AMI, is a resource and initiative for Africa, but it is hosted and supported by the University of Cape Town and the Institute of Infectious Diseases and Molecular Medicine in Cape Town. And the equipment that we have in the lab is sourced and supported with extensive training for users from Zeiss and Tissue Gnostics. And they have committed ongoing support for the initiative for support of their equipment, for software licenses, as well as training of users on those systems, which is very generous of them. We also cannot function in isolation from the rest of the world communities. So we have strong commitments from Eurobioimaging and France Bioimaging to support development in Africa. It's valuable from France Bioimaging because of the large Francophone community in Africa. And that is also possibly a space that researchers in Montreal could get involved in as well. And there are funding initiatives that France Bioimaging has already announced that will facilitate African researchers accessing French imaging infrastructure where needed. So that's very generous of them. And obviously, researchers get connected to the African Bioimaging Consortium. And as I mentioned, we're generously supported by CCI and the Gates Foundation. And once again, I have the privilege of standing up and telling everyone about this, but it doesn't happen in any way with just one person's efforts. So just to acknowledge key team members being my colleague, Michael Reiche, who will actually be the operations manager for the imaging center back in Cape Town. Prof. Warner and Ms. Rahi in Cape Town are incredible advocates for all of our efforts. And Dr. Teng Yong Chu from Geneva AIC has been an advocate and driving force in making sure that all of this comes to fruition, and will be serving on the AMI advisory board going forward. So I wanted to end with just highlighting, reiterating where those little light bulbs were, that there are ways for the international community to get involved in everything. One of the biggest and simplest ways is word of mouth. If we have all of these initiatives developed for researchers and the African community does not know about it and cannot take advantage of it, it is unfortunately all for naught. So please, if you have any colleagues, collaborators, or students that you think might be interested, even if it's because you think they might know someone who might be interested, please share the word. There are trainers needed for the application writing workshops that we're planning to run through ABIF. So if anyone's interested in that, please get in touch. There will be mentors needed for imaging scientists and early career researchers. For any early career researchers in the audience, please think about applying for pitch when the call goes out. And we're also looking for advisors for that RC expert for a program. So once again, get in touch if that sounds interesting. And seeing as I've already kind of given acknowledgements, I'll leave this slide up for contact details for both of the initiatives I've spoken about today. And I'm happy to take any questions. Questions?